Why do we need to collect specimens? Why do we need collections and museums? So it's kind of a very broad topic, but of course, I'm not going to cover everything. So if you have questions, please keep them, and I'm more than happy to answer any that you may have. Maybe at the end of the talk, it may be easier this way, or maybe after the end of both sessions. So first, I'd like to start with the questions for you. Do you know simply how many species are found on Earth? So that includes everything: animals, plants, fungi. Let's leave bacteria and other like microscopic organisms aside, but let's think about all of those together. Does anyone have an idea? Thank you. It's good. I see people in the chat. Yes. Or you can just think in your head what you think would be the answer. How many do we know?
Maisie, do you know? Just give a number.
Is it more than 10?
Oh. So Mavis have an answer. Is that too big?
I have a big number here. Yeah. Oh, it's like 20 billion? That's really a lot. Okay. Well, the answer for me is that we actually don't really know. But we have guesses. And I will try to explain to you why we don't really know, but what we know. So it's always something I'm interested in. And first, I'd like to focus on what we know. So here we have a map of the Earth. I'm sorry, it's not the most beautiful map, but I will explain in a few minutes what it is. And first, I want to introduce to you how many species we know. So that means science has described the number of species. And currently, we know about 2 million species. Okay, 2 million, that's already quite a big number. We can all agree on that. And what you can see here is on my map, I actually have different colors. So now I'm going to try to explain to you what are those different colors. So let's imagine that every species, big or small, whatever they are, they will occupy the same area on the Earth. Okay, so you take the overall area of Earth and you divide that by 2 million, and that gives you the area occupied by each species. And then we sum up this area for all kinds of groups of organisms. And that's what I want to represent here on the map. And the first one that will come are actually insects. So imagine that we have all of this. That means insects will actually be covering about 56% of Earth. That's absolutely gigantic. And that's because insects are by far the most diverse group that we know of. Then if we had like the other ones that are somewhat related to insects, which go into a group called the arthropods, and so that includes things like the spiders and scorpions for the arachnids, but also the crabs. So those are mainly more marine, although there's a few terrestrial crabs as well. And then we have a group called the Myria Pods, which are basically the centipedes and the millipedes. You can see that all of those together occupy about 2/3 of the Earth's surface. So they're really like the most species-rich group that we find. Then if we keep going with the rest of the other invertebrates, so we have groups like the mollusks, for example, which are the snails. We have some nematodes, annelids, and platenmen. Those are more like worm-like kind of animals. And then we have the bryozoa, which are the corals. You can see that if we add those to the arthropods, so all invertebrates, that's about 3/4 of all life on the planet. Then let's focus on other very important groups as well, the plants and the fungi. We can see that the plants, for example, will be covering all of North America and even going as far as Central America. And then we have the fungi, which will be covering most of the Amazonian basin. And then finally, there is us, the vertebrates. And here you can see that actually when we talk about vertebrate, half of the diversity are fishes. And then the rest are the birds, the amphibians, the mammals, and the reptiles. And thus, in terms of diversity, they are actually not so diverse. And I'll give you the details just in the next slide. But for example, the birds, we're covering France, which is where I'm from originally, and a few other countries nearby, which, you know, maybe French may not like it, but that's a pretty small country. So let's be very modest here. Now, If we look at the diversity of life in terms of numbers of species, that's what I show you. So now I ranked, you have this table where I ranked everything, and I put a little picture beside each other, so you can, each name, so you can have an idea of what they look like or what they are. And you have those numbers. So again, insects, that's about 1.1 million species. I told you this is more than half. But then we also have to think about how many species we do not know yet. And that's quite a very open question and a bit difficult one to answer sometimes. But people have come up with different estimates. And here I show you those different estimates here on this new column. And you can see that there are clearly some big differences in those numbers. For example, with insects, we know about 1.1 million species, but most estimate now will be around 5 million. Sometimes think there's 30 million species, maybe we can find 100 million species. That's maybe a bit of an exaggeration, but I think most people would agree maybe 5 million seems like a reasonable number. That doesn't mean they're right, because that's a primitive estimate. Now, if we look into detail with those groups, we can really separate three groups here. First, we have those that are relatively well known. So those would be the groups for which we know almost everything, or we assume we know everything. Groups like plants, we know about 80% of the species, which is quite good. Of course, it still means that there is 20% to discover. But some other groups and most groups of vertebrates in particular are relatively well known. Like even for some groups like mammals, we pretty much know all of them, although I can discuss later on, maybe there's some limitation here. Then there's a second group for which we know half to maybe 2/3 of the species. And so those are amphibians, for example, which is a group of vertebrates for which we have the least knowledge in terms of the species diversity. So there's still a lot of work to do, and I'm sure we're going to be fascinated by hearing more in the next talk. But you also have things like the crabs, for example, or the mollusks. And then there are those for which we don't even know a quarter of the species that may exist on the planet. And here we have insects, arachnids, so the spiders and relative, all the mushrooms, fungi, but also the groups like platyhelminths and nematodes and the Myria Pods. So that means that our current knowledge of all those groups is very limited. Here, usually I use this slide for insect diversity, and one of the things that is marking, because insects are so overwhelmingly represented here, we have 80% of them that are still unknown, and in parallel, we estimate that there's about 80% of species on the planet that are unknown. This is a staggering number if you think about it. That means that in 2022, we only know 1/5 of the species on this planet, meaning that 4 out of 5 species are still unknown. As a biologist, sorry, I can tell you that I'm very surprised that we are so eager to spend billions and billions to explore life in other planets, but we don't know even 80% of the species on this planet. And we know that many of them are actually disappearing, and before we can even discover them. So I think we might have to think a bit more for the future where we want to invest our money, because so far, as we know, the only life is here on this planet. Now, we have to consider that when we talk about biodiversity, we actually talk about something that we mainly don't know. That's the unknown majority. And that's what I want to start with, to show you how is it important to actually collect organisms. And when we do that, we can do that in all kinds of ways, in all kinds of places, and in all kinds of habitats. So you can be an entomologist like me, working with insects, and then means you might be collecting maybe ants or butterflies or other things with a net. But you can be working in freshwater, for example, like if you work with amphibians. You can be working on the canopy, so on the top of the forest, and this is the top middle picture here, where you can see what is known as the canopy raft. And so people can collect plants, but also fungi and insects, and even vertebrates. There's a lot to discover there. You can walk under the ocean or on top of the ocean to discover new kinds of life on the beach. And of course, it's something that scientists do, but also the younger generation can contribute. And that's very important. And then when you grow up, you can keep doing it and contribute to all this knowledge that accumulates all over the world. But some of the most striking example to me of how diverse life is coming from one scientific study that was done about 10 years ago, or completed 10 years ago, where a group of scientists decided to actually study all the insects and other related organisms, so spiders and so forth, the arthropods, in half of an hectare of a tropical rainforest in Panama. So half of an hectare is actually not a big area. That's about 50 by 100 meters long. And then they actually found 6,144 species just in that half of an hectare. It took them 10 years of work to identify everything. Just to give you an idea, in that half of an hectare, there was actually more species of arthropods than there are mammals on the entire planet. So that shows you like how diverse those are. And to me, this is very heartbreaking when I hear about deforestation, because that means there's so many species that are being swept away with that. And just to give you an idea, they collected over 130,000 individuals. That's a lot of bugs here and related organisms to collect. So the question is, why do we need to collect all those specimens and those samples? And that's what I want to focus on now. Now, we know, for example, some studies can have a lot of them, but why is it really necessary to do that? Well, there are many reasons for this, and I'm not going to touch on every single one of them, but probably on 4 that I think are the main ones. The first one is for identification. So if you maybe observe birds or butterflies, you may know that sometimes you may just need some good eyes and maybe a pair of binoculars. And for some of those groups, observation in nature is good to get an identification. But unfortunately, for most of the life, this is not enough. We cannot identify species by just looking at them flying or running by. But even for birds and butterflies, we realize today that many of what we thought were good species can actually be multiple species. And that's what we call cryptic species, where you can have multiple species that look exactly alike, and you need to do then more analysis. And for doing that, you either need to collect some samples like DNA, for example, or you need to take measurement on the individuals. And so that will often require either a sample, but most of the time it will also require to have specimens. And for those other groups, most invertebrates, but also some plants and fungus, mammal groups, for example, if you work on rodents, you will often need to have specimens. They require, again, samples of specimens. And so that's what you need to collect specimens. And myself, I work with ants. And here you have some examples. So once we collect the ants, we mount them on pins, which is what you can see on the top left here of your screen. Then we can have collections, which are in box of specimens that looks alike. And then we look at them through microscopes, different microscopes. Sometimes we can look at them directly with the microscope and look at different characters, and I will explain that later. But sometimes we also have to image them to actually focus on particular parts of the body and make sure that we can really observe some very fine detail to validate the identification. And I will focus on one particular genus of ants called Genus. So ants are what we call a family in insects, and there are multiple genera that are known around the world. Actually, we know over 345 genera at this point. And Genus is a very interesting one to me because they live in the leaf litter or in the soil. And here you can see an entire colony on the picture on the left. And probably, I'm not the person who took that picture. This is one of my colleagues called Alex Wild. But probably like the entire nest is about 2 centimeters in diameter. Because Genus are very small ants. They measure 1.5 to maybe the largest one, 5 millimeters, with most of the species measuring about 2 to 3 millimeters. So it's very, very small. You can think about how big that is. And here you can see one of the species, for example. And if you look on the bottom right, you have the head of that species. And if you pay attention, you have a scale, and you can see that the scale bar is actually 0.1 millimeter, so 1/10 of a millimeter. And so the entire head here is probably about 0.3 millimeter. So that we're talking about tiny, tiny ants. But they do something amazing, is that they're often predators of springtails, also known as columbola for scientists, which are other arthropods that live in the leaf litter, and they use what is known as a trap jaw mechanism. And to do that, what they will do is that they have mandibles on the top, just at the extremity of the head, and they will open them, sometimes up to 180 degrees, and they will close them very fast. It's actually so fast that it's one of the fastest movements in the entire animal kingdom. So it's very, very fast movement. And with the shock that they create, they will be able to collect those insects like springtails, who also are very good at jumping and escaping very fast. So that's why they have this movement of mandibles that are very fast, so they can get a better chance to collect those insects, which themselves are very fast. And one other thing that is amazing is it is an hyper-diverse groups. In that single genus, there are nearly 900 species described. And so, of course, when you collect one specimen, you cannot tell right away, this is this one or this one. You have to consider that potentially it can be any of those species. And that takes time. And of course, you need the specimen to do that because they are so small to actually get all those details. And here in 2018, one of the students in my laboratory, Wilfried, approached me and he said he was interested to do a revision of the species in Hong Kong. And we asked a very simple question. How many species of Strumigenes can be found in Hong Kong? So we didn't start with a blank slate. There was actually 12 species that already had been recorded in Hong Kong. But we decided to go back and with all the collection that we have been doing over the previous 4 years, and we start looking at those species. And here you can see different heads of 12 different species of Strumigenes. And I hope you can see the difference between some of them. Maybe not all of them, some, especially on the left column, they might look a bit similar, but you can see that some have long mandibles, some have shorter mandibles, some have mandibles that are triangular, others that are more like elongate and more skinny. Some of the head have a lot of what looks almost like light bulbs, which actually have some modified hair. Others are very fine hair. So they are quite different. And you can only perceive that once you look at them under a very good microscope. So thus you need the specimens, of course. And once we do the revision, we actually found that there were 24 species in Hong Kong. So clearly we started with a past record of 12 species, we doubled that number. So it was much more diverse than what we thought it was initially. And one of the things Wilfried also realized is that some of the species were unknown to science. And actually, during his work, Wilfried described 3 new species to science. So he was a young scientist in his career, and he had the chance to describe 3 new species, which is great because that's something that I think is a legacy for the rest of humanity. Those species will probably, hopefully, will survive and will be around for many, many more years in Hong Kong and other places around the world. And just very recently, actually just a few weeks ago, we discovered what is the 25th species in Hong Kong. And this one, you can see, it looks very different from all the previous one. And it's also a new species for science. And it's found from a place we will not guess that a new species could be found there. It's kind of a disturbed area, not so good, not where we imagine new species to be discovered. And yet that's where we found it. So now Wilfried is also working on a larger collection that we had. So we collected specimens from Hainan in China, from Vietnam, from Thailand. And guess what? He found new species as well. And he's describing 15 new species to science. He actually just sent me the latest version of his manuscript last night. So there is a lot of new species to be discovered. And those can only be described if you have the specimens to really make all kinds of observation. You cannot do it just by seeing a specimen in nature. But the case of Wilfried is not the only one. Here is Ying, who actually discovered a new ant species just about 200 meters from our laboratory. Just one night, she goes collecting ants, and she found this new species, which is known as the golden tree ant in Hong Kong. So she has one new species. Here's Danny, who described 3 new species from Macau. If you've been to Macau, you may know it's not exactly the most friendly place to discover what you would think for biodiversity. And yet, Danny found 3 new species over there. It's very disturbed, but there are still new ant species that and other insects as well that await to be discovered. And here is Daniel. Daniel was an undergraduate in my laboratory, was his final year project. And he described 4 new ant species from Hong Kong, and now he's describing 5 new other species from Southeast Asia. And I just want to focus on Daniel a little bit more here, and exactly part of his project. Because this project actually did not start in 2020, but it started earlier. And it started with this gentleman here, Dr. John Fellows, who studied ants in Hong Kong in the early 1990s. And what John did then was that he had a very brilliant idea. All his collection of ants, he decided to keep it. So he kept every single specimen he had. Either he mounted them, just like I showed you earlier, or he kept them in ethanol, so in some kind of alcohol to preserve the specimen. And that was really brilliant. And he deposited his collection. He deposited at Cadbury Farm and Botanical Garden, for those of Hong Kong who may know the place. And this collection since is being hosted by the Hong Kong Biodiversity Museum, which I will introduce later on. And Daniel, when he came to see me, he told me, oh, I'd be interested in studying the ant genus called Polyrachis in Hong Kong. And then Daniel and I, we had a brilliant idea as well. We said, hey, you know what? Why don't we go back to John's collection and see if there is anything interesting that we can find in this collection? And so Daniel did this, and he identified 17 species of Polyrachis in Hong Kong. Before his work, we only knew 9 species. So that means we discovered that actually there were new records, and there were also new species for science, 4 exactly. And one of them was actually a species that was collected by John Fellows in 1992. And then we decided it would be great to actually name the ant after John. And so that's why this species now is called Polyrachis Fellowesi, which is basically the Latinized name of John. And so he was very happy when we told him that. He felt like, you know, it was great that all his efforts in the early 90s were rewarded with the species that he discovered. Didn't know then, but then was described after him. Now, when we have specimens, there's a lot of other things we can do. And in particular, we can do ecological studies. And we can take a lot of measurements. So here I'm just showing you like all kinds of measurements we can take on the head, on the mandibles, on the antennae, even on the body. All kinds of different parts can be measured. And that's what a lot of scientists have been doing traditionally. But as technology progresses, there are more things we can do. Now we can actually obtain an entire CT scan of an ant, just like, you know, you can have a CT scan if you're sick, which I hope you won't be sick. You go to a medical facility and they might have a CT scanner over there. Well, now we can do that with all kinds of life forms, including small insects like Genus. And not only we can see details on the outside of the body that before we could not perceive, but we can also see inside the body. So that gives us a new dimension to work with. And when we have all those measurements, we can do some particular studies. There's a lot of things we can do, but here I'm just going to be focusing on the change of multiple species in function of their habitat, maybe in function of disturbance, or in function of time. And that's one of my former PhD students, Dr. Roger Hawley, wanted to do. We wanted to study the impact of climate change and disturbance on ants in Hong Kong. And so what we've done with Roger, we asked ourselves, okay, how ant community change over time? Do they change in function of the climate? Do they change in function of the habitat? And we wanted to compare the modern communities. So Roger went to do field work and he collect ants. So here you can see him collecting leaf litter and all the insects in the leaf litter. But he wanted to compare those with past collection. The problem is we don't have a time machine to buy on eBay or anywhere else. So how do we go back in time and are able to do any meaningful comparison? Well, fortunately, we had John again when we had his entire collection. We had every single specimen he collected. We knew exactly where he collected them. So what Roger did, he went back to the same sites, he collected the modern communities, and then he could compare them directly with the collection of John that was created 30 years before. And that's the collection of Roger. So he had those. We could do direct comparison, and we could compare the body size in particular. And then we compared what happened in terms of climate in the 1990s with what happened in the mid-2010s. And here, just a few figures like that to show you, but what you can see is the number of extreme hot days. So it's a hot day in Hong Kong is defined as a day when the temperature goes above 33 degrees Celsius, the air temperature. And what you can see, unfortunately, on these figures is that since the 1990s, the frequency of hot days in Hong Kong has tripled. So where you used to have maybe 10 per year in the 1990s, now 20, 25 years later, you get about 30 days every year. So clearly that means that the organisms, animals and plants that live there are experiencing much more warmer temperature more frequently. And we were curious to see if there is an impact on that, on ants in particular. And what we did find is that yes, there was one. And this effect was for body size to decrease. So the ants in the 1990s used to be bigger than the ants in the 2010s. So, and that seems to be directly related to temperature. We tested other variables, but there was many temperature. But what was very interesting is that this effect of body size decrease was only observed in open habitat. We did not see that in forest habitat. And that makes sense because if you are under a forest, you're under the canopies of trees. And that canopy buffer protects you against the very hot temperature. And what our results actually suggest is that one of the best ways to fight against climate change is also to limit deforestation, to make sure we can preserve vegetation, and in particular, older vegetation, which have a much more complete canopy. The branches are providing a lot of shade, and that will buffer the impact of climate change. So this is very important, in particular, within subtropical and tropical regions. Now we are starting a new project where we're not looking at ants this time, but we're looking at bees and wasps and how they change in Hong Kong over time. And this project is being led by an undergraduate student, Clara Park, with the help of one of my PhD students, Rangzi Wong. And all the data were collected by Christophe Barthélémy. So Christophe is not a scientist. He used to be an architect. Now he's a farmer, but he's an amateur entomologist. And Christophe is a specialist of bees and wasps, all kinds of them. And he has this thing for 18 years where he keeps sampling bees and wasps all the time. And he kept all his collection at home, which is great. Again, keeping those specimens is so important. And when he did that at multiple locations, but at one location in particular, for 18 years, he sampled bees and wasps continuously for 18 years. Every 2 weeks, he was going and collecting them using mallet straps, which are the straps you can see in the bottom left here. And this actually a trap where the insects will go underneath in the black part you can see, and then they will start going up, and on the very top they will fall into a big valve that contains ethanol, so you can actually collect them. And you can see the change over time. And here on this figure, you can see some of the change in species richness, which are the blue lines. So you can see sometimes the line goes up, sometimes it goes down, and in abundance, the numbers of individuals that were collected here as well. And you can see that there is a lot of variation going on. And so what we are interested now is to understand those variations. What actually is driving them? Is it maybe the climate and the different part of the seasonality and temperature, precipitation, etc. So this is something that we're just about to start. And in particular, what we'll be doing is to also measure the body size of those different individuals, but also something that we call filtrating asymmetry. I'm sorry, that's a bit difficult term, but basically during the development of an organism, when it basically goes from the young to the adult stage, sometimes if they are under some stress, they don't develop very well. So maybe one wing may be a bit bigger than the other one. And so that's also one of the things we're interested in measuring to see if climate may have an impact on them. So I don't have any results for that because we are just starting that project. That's something we're really excited to look at. And then there is for conservation. Keeping specimen is very important for conservation. I think most of you here might be too young, but in 1990, when Jurassic Park, the very first one, I don't know how many there are now, maybe 4 or 5, came out, well, this was really like spectacular. And if you've seen that one, you may know the story. Is that kind of rich collector slash scientist who found this piece of amber with a mosquito imprisoned inside, and then he extracts the DNA from that mosquito and is able to recreate dinosaurs. And back then, when it came out, it was completely science fiction. But now, it's not so much science fiction because people are thinking about doing it, not for dinosaurs, but for other extinct organisms, such as mammoth. And so it's not clear yet if it's going to be a success, but it's definitely something that is in the realm of possibility. And so by preserving specimens, we actually are able to have some kind of a reservoir on which we can extract DNA from those old specimens, and then potentially keep giving them an extra life over time by resurrecting them as a species. That's also important for preserving all that genetic diversity, because when we talk about a species, it's not just one individual, but there are multiple populations, and all of those have different genomes, and we want to preserve all of that as much as possible. And all of this can be important for the conservation of species, but also for agriculture. You may know that there are different kinds of plants or different kinds of domestic animals. They all have different values, and some of them, and a lot of them, unfortunately, are also getting extinct. And preserving those specimens is actually ensuring that in the future, especially as we have a world that is changing very quickly, we might be able to find either the right kind of plants or the right kind of animals to support our own societies. And it's the same for medicine, because there's probably a lot of remedies that we can extract from plants and animals that if they go extinct, we lose everything. So, but how do we keep all of that? And that's the second part of my talk, which is about preserving that natural heritage. And that's where natural history collections are so important. One of the main functions is, of course, to educate the public about the richness and importance of biodiversity. So here you have this absolutely amazing collection. The buildings are also as impressive as the collection themselves. But it's really a great way to educate the public. And I think one of the things that we should never forget, it's just to be amazed by the diversity of life. Really to ourselves think like, wow, how those things are so amazing, so beautiful, so interesting. So we are just curious to learn about them, and we are curious to actually want to protect them after in their natural habitat as well. But the collections themselves, to me, I see them as biobank. So they are preserving life so we can understand and protect it better. And this is very important to preserve all this life, even if it's with specimens, so we can basically know more about them and understand how they change over time, just like in the example I showed you earlier. But we can also foster the new generation, maybe you as the new scientists will be studying them, or later as citizens who would like to protect them. And it's also a great way to communicate with the media. The media love those collections. It makes some nice image for them, and that allows us to reach out a larger group of people in the general public. But here's something that we really need to improve. Now, I told you earlier about biodiversity and how many species there are. The thing with biodiversity is that it's actually not the same numbers of species all around the world. And here I just took 4 examples for which we have data. Those are mammals, the birds, amphibians. And very recently, we just published this article about ant species richness around the world. And what you can see here is that the warmer the color, so the more red it is, the more species you have in those areas. And the opposite, the blue, the colder or the bluer it is, the least species you have. And you can see that the majority of species are actually found within the tropical region. And this is where Hong Kong is, for example. But then if you go further south of Hong Kong, you will find even more species there. I show you for 4 groups, but this was another paper that we published a few years ago, showing different groups of plants as well for freshwater, marine, etc. And many, many groups, actually most of life, will show this pattern. There are a few exceptions, but most will show that pattern, where most of the majority of global biodiversity is located within tropical regions. But unfortunately, very few museums and very few collections are found within the tropical region. And this is a problem, because that means that the scientists who want to study them in those regions have to travel to other regions. And the best thing, I mean, I've been in Hong Kong for 8 years now. I cherish every opportunity I have to just go outside and study those specimens whenever I can. And it's very different when you only there in a region for a few weeks or a few months. And so that's really also giving great opportunity for the local scientists, but also those who might be traveling there to get facilities available. So I'm certain that by having more facilities in the tropics, it will actually increase our understanding and knowledge of the different fauna and flora that are found in those regions. And so that's why we need more museums where biodiversity peaks to speed up research, but also education. And that's very important because we need to educate people in those regions by showing them, look, you have so many things that you can be amazed, that you can appreciate. And I think museums are a fantastic way to do that, and then increase the protection, because knowledge leads to better protection. And when I joined in Hong Kong in 2014, one of the things that surprised me, actually many things surprised me. One is Hong Kong has a lot of banks. It also has a lot of museums. You may have heard there's a lot of great, fantastic museums, the M+, the Heritage Museum of Hong Kong that have opened recently. But one thing that really surprised me, there was no natural history museum. And then I thought this is something that has to change. And that's why I decided to use the collection, small collection, very modest, found within HK University of Hong Kong to create the Hong Kong Biodiversity Museum. And it's probably one of the smallest museums in Hong Kong. We only have 62 square meters, but we have a collection of 20,000 specimens, which again is very, very modest. Most large collection, we have several millions of specimens. But nonetheless, we just opened recently, first online, and I'll be talking about it in a second, in 2021, and then physically to the visitors in May. And we welcome 9,000 visitors. So if you did not visit us yet, tell your parents, I want to go there. I want to see that museum. It looks so cool. But we also, because we are a small museum, we have to be creative, and we try to be present online. And so every week, we'll actually introduce a particular species or particular concept of biodiversity. And so far, you have over 135 stories that you can read and learn more about all kinds of animals and plants. We already have 8,000 followers, and we have reached about 400,000 people. So we are really happy about that. So you can just click here and get access to any of our social media here. And we really talk about anything. We talk about arthropods, vertebrates, terrestrial, marine creatures, plants, all kinds of things. We really try to talk about one thing that is important to us is to teach and have people learning about local tropical biodiversity, because that's where you live. That's the species that you interact. And we want you to be amazed by them and want to learn more in the future and protect them. So this is the end of my talk. So I would like to thank the different funding agencies that we have. And I'm very glad that 2 days ago, we get a renewed funding for the museum. So for the next 2 years, we know now that we could remain open and continue our activities. And also Aline Oliveira, who is a curator at the museum, and Anders Yu, who is our education manager at the museum. And because we're a small structure, we have the luck to have a great team of interns and volunteers who are helping us on Wednesday and Saturday when we are open. So it's really, really helpful. And thanks again, Benita, for the invitation. I hope you liked the talk, and maybe you will have more questions later on. So thanks.
Thank you, Benoit. So yeah, any student have questions? I also want to ask, so I saw you have some young student like Wilfred, who is like discovering the ants. So what can students at this age, other than going to the museum, how can they contribute to biodiversity? I don't know, using the iNaturalist or whatever.
Yeah, they can start learning. I think it's never too early to learn. And I know in Hong Kong, for example, for ants, there are some young teenager or even kids who actually are getting pretty good knowledge of just collecting and being curious. I think, be curious about what is around you, and whatever the tool that you use, either iNaturalist or buy a book, or maybe try to get a small microscope if you can. There's some that are relatively cheap. Just be curious, observe, or just a lens that can grow 20 times. if you are more into the small things, the plants are very easy to observe as well. So there's a lot of interesting plants that can be developed. There's all kinds of books, and I think it's really a step-by-step process. The books that I use, for example, or my students use, they are definitely way too advanced. But there are some where you can learn, a little bit by little bit. And contact researcher. We had students coming as young as 15, 16 years old, and they have spent like multiple summers with us doing projects. Just yesterday, I was talking to one of my former PhD who now has a position in England University. He's going to start working with 16 years old. We're going to be doing a project together. We try to make them like custom made, but it is possible. And we coming with the museum is also, of course, a good thing because they can talk more about us, and we can directly see what kind of interaction or collaboration we can do. And I just want to finish by that. There's a lot of citizen science projects that exist. Those are a very good getaway to start doing some very exciting research here.
Thank you. Any question? Kate, Jaden, Mavis? Afina? Okay. So if we'll go to Jennifer, and if you want to ask again, please keep it in the chat room, or you can unmute yourself. So your time now, Jennifer.
All right. Unmute myself. I think this is my slideshow. No. Are you seeing the slideshow? Or okay, give me 1 second to figure this out.
Yeah, that works.
Sorry.
That works. Just now it works.
Oh, okay. Sorry. I'm like an unfrozen human. Hang on 1 second. Sorry. Share slideshow from beginning. How about that?
Just don't move it. Let it load. It was loading just now.
Okay.
Okay, that works.
Great.
Perfect.
Okay, so I am going to talk about why we collect amphibians in particular. First of all, Benoit, I loved your talk. That was really great. And I love that you touched on a lot of the same things, but I'm going to touch on from a different perspective. So I was so happy to see all of the things that you were talking about in there. I love it when people talk about how great collections are, and it's great to hear kind of these same themes repeated from different taxonomic perspectives. So I am an ecologist and a herpetologist. An ecologist is someone who studies how organisms relate to each other or to their environment. A herpetologist is someone who studies amphibians and or reptiles, and I mainly focus on frogs. And I am the curator of amphibians and reptiles here. This is the Carnegie Museum of Natural History. This is located in Pittsburgh, Pennsylvania. And many people know of this museum because of this very famous dinosaur. So when people come to our museum, they often think of this as the Carnegie. This is Diplodocus carnegii, and we call him Dippy for short. But again, most visitors that come to the museum, they think of dinosaurs, which is what they see on display. But what many people don't know is that this museum and every other museum has extensive collections behind the scenes that most visitors don't get to see. So this is what we call the alcohol house, which is where the amphibians and reptiles are stored. So as Benoit showed you with insects, those collections are often pinned. They're dry specimens that are pinned, and they're in boxes and drawers. With amphibians and reptiles, the majority of those collections are held in jars of ethanol. This is the way that we preserve amphibians and reptiles for the most part. So we have over 250,000 individual specimens in our collection. And again, most people don't get to see this. But the reason I'm showing you this is to show you kind of like the breadth of this collection, to emphasize, sorry about this background noise, emphasize that most people don't get to see this, but it's also a living collection. So as Benoit, 1 second, please. Sorry, neighborhood noise. As Benoit talked about, we use these collections on a regular basis to answer different questions. So all of these collections here, we get visitors coming into our section who are looking at taxonomy. They want to describe new species. We use them to answer ecological questions. We get loan requests. So we give our specimens to a researcher who wants to study them. And so people can either come to us or our specimens can go to a researcher. And so we have all of these specimens. And one of the reasons that we make so many collections, a question we often get is, why do we need more collections? So if we have all of these jars already, then why do we need more? We've already taken all of these things out of the wild. Why do we have to keep collecting? And one of the answers to that, and I'm going to talk about many answers to that question, but one of the answers to that is that these are really a snapshot in time. So as Benoit showed you with that, the collections made by fellows in the 90s, that was a snapshot of what existed in Hong Kong in the 90s. Now, that is not to say that that will never change, right? So as they showed, we know that diversity is changing over time. We know that things like size is changing over time. There's lots of different things that change over time. And so by taking regular snapshots over many, many years, we can then understand how our world is changing and how diversity is changing. So I focused on frogs for the most part. These are all frogs from Borneo. That's where I currently am. And I'm going to talk a little bit about what I'm doing here at the moment. But this is just to show you one of the reasons why I love frogs. I just think they're so cute. Most of these are tree frogs, which are very photogenic. They've got big eyes and big sticky toe pads. I love the grumpy guy in the center bottom, Magophrys nasuda, which looks incredibly like a leaf if you were to see him from the back. They've got just great personality. So I study frogs and I look at the impacts of land use change and the impacts of climate change on this group. And as we talked about before, one of the reasons that we take specimens is to document diversity. And so this is me in the field in Laos. So when I was a postdoc at the museum in North Carolina, my advisor was Brian Stewart, and he worked largely in Laos. And we went to Laos a few times and we made collections. This is an example of what it looks like. So the photo on the right is how you set up these amphibians in the field. You fix them in formalin and then you store them long-term in ethanol. And as Benoit touched on, there's often what we call cryptic diversity. So we know many species, but as he emphasized, there's lots of species that we don't know. And with amphibians in Southeast Asia, this is particularly true. So there's lots of new species described every year. And as he mentioned, it's not necessarily things that you see and it's so different from everything else that's known. Usually it's something that you think could be something that's already described, but you're not sure, but it looks really similar. So one of the things that we did with that collection is we described several new species from across Southeast Asia in this group. This Hylarana nigrovittata at the top was already described and defined, as was this one on the right, Hylarana mortensini. And these other species here are ones that we described as new. And it relied on collections, not just that we made, but other collections from many, many museums made by many, many researchers over this huge geographic area. So originally it was thought that these two species had really broad geographic ranges. So nigrovittata was represented by these black dots and mortensini by these blue squares. And so these were thought to be really widespread common species. And what we found when we looked at the genetics, when we looked at morphology, we found that there were actually multiple species in here. And this really changed what we thought of as the species distributions for these things. So as Benoit emphasized many times, which I loved hearing, was that you can't always define these things just by a photograph, for example. So that we often get this question of, why can't you just take photographs? Why do you have to kind of take these out of the wild? And why do you have to make specimens? And as he mentioned, you can't always get all the information you need from just an observation, from a photograph. You need that whole specimen, whether it's the genetic material or whether it's looking at the morphology. So we looked at the genetics and we looked at the morphology, and we were able to define these species. And in addition to just defining diversity, one of the reasons this is important is for making educated and informed conservation decisions. So you can imagine if you are a land manager or a planner and you're trying to figure out, you have a limited budget, you need to decide, what are the regions or areas that I want to preserve? Where do I want to kind of keep primary forest habitat intact? Well, if you have this map on the left and you're making your decision based on this map, you might say to yourself, well, this species represented by the black dots is really widespread. So if I just lose this one population over here, it's not going to be a big deal because there's so many other populations of this species. But it turns out that now that we know what the true diversity of this species group is, that one area is actually the entire known distribution of this one species. So by having these descriptions and by updating our taxonomy, by using these collections, we can make better conservation decisions because we have a better understanding of diversity and species ranges. So again, this is only possible by using museum collections, by making new collections, and really understanding what that diversity is. So these museum collections are really important for conservation, for making informed conservation decisions. Another thing that I want to talk about is what I'm doing here now in Borneo. And what I am working on is a project looking at the differences between diversity and productivity, which is basically energy in a system, how those two things are related across land use type. And this is also an important reason for making collections. So I'm currently working in primary forests, secondary forests, and agriculture. The experimentally fragmented forest is part of this bigger project, but this summer I'm focusing on primary, secondary, and agriculture. And what we are doing is we're going out at night and we are looking for frogs on these streams, and we are measuring them, we're marking every individual, and we're weighing every individual. And we're looking at differences in diversity across these streams. And we're looking at differences in biomass. So is are they fatter in primary forest, or are they skinnier in secondary forest? Are there differences in sizes based on their habitat? And one of the reasons that we make collections as part of this work, so this ecological field work, not every ecologist does make collections, but we do. And one of the reasons is that here where we work, so I'm here in Sabah, and I am in this area called the Crocker Range. So in this area, there's a lot of groups of amphibians that have this cryptic diversity that we've been talking about. And one of those groups is this Marista Genus. So this is a Arachnid. It's kind of a very common frog type thing. The photo on the right is an example of a species on one of our streams, and we don't actually know what it is. So it's most similar to this defined species called Stigmachilus, but it differs from that species in a couple of key ways. So in order to understand what the diversity is on the streams where we're working, we're making collections, we're making specimens to represent what it is that we're seeing, so that we can define, okay, here's the diversity that we're seeing. We know for sure what the species is. We make those collections so that after my field season is over, we can compare our specimens 2 specimens of this species, Stigmachilus, and see, okay, is it actually that species or is it something else? And so this helps us define our species. There's another stream here, same genus, where we have this guy, looks very similar. It looks a lot like Stenocephalus, but it differs in a couple of key ways. You can see that this is hard to see in this photo, the inside of this tympanum or the eardrum in the Stenocephalus is dark, where on our guy, it's very light. And this is one of the things that defines this species. So we're making collections so we can later again compare it to known specimens of Stenocephalus to say, okay, is this what we have, or do we have something that's yet undescribed? Or is this just natural variation in the species that is not yet known? This species has been described from only a handful of specimens. So it's possible there's just much more variation in the species than was previously known. So in ecology and types of work that I do, like I said, we make these collections to help know for sure what is the diversity that we're seeing, and that helps us answer this bigger question of how is diversity related to productivity or energy in a system, and how does that vary across land use type. And another big question, similar to Benoit's work, that I work on is how organisms are related to climate change. So we know that the climate is warming, we know that precipitation patterns are changing around the world, and we know that these factors, increases in temperature and changes in precipitation, have affected organisms in a lot of different ways. So one thing we know is that phenology, which is a fancy way of saying breeding or flowering, we know that breeding and flowering have been impacted by climate change. We know that organisms have shifted their range in response to changes in climate. So often what we see is that organisms do attract their thermal niche. So they say, well, this area is a lot hotter than it used to be. I don't like it here. So they go to where their thermal niche, where their preferred temperatures are. And we know that this is happening. And as Benoit touched on, we also know that body size is changing. So this has been shown broadly across many species. We generally see that organisms are getting smaller in response to warming temperatures. And all of these studies have been enabled in large part by museum collections. So as Benoit said, we don't have a time machine. We can't go back in time and look at them, but these museum collections serve as that time machine. So we have museum collections around the world. We can look at species from different places in different decades, different centuries, and we can compare today with what they were decades and centuries. And recently I looked at frogs in Borneo. I got my hands on every specimen from Borneo that I could find from many different museums around the world, both here in Sabah, as well as the British Museum. There's a museum in Chicago called the Field Museum that has a great collection from Borneo and several others. And I looked at body size of multiple species and looked at how broadly these frogs are changing. And what we found was really interesting, that in general, they're getting smaller, but that changes in precipitation exacerbate that change in size. So there's what we call an interaction between temperature and precipitation, that when places are getting wetter, the species get much, much smaller than when a place is getting drier. So in dry environments, they're not declining as much. In wet environments, they're getting much, much smaller. And again, this study was enabled by extensive museum collections. So we had over 3,000 individuals, all in museum collections from many, many places. And this is only possible from continued collection. So again, this comes back to that question that I get all the time of, well, you have all these specimens in jars, isn't that enough? And the answer is no, because we need to keep documenting diversity. There was a great paper that came out recently that showed that actually new collections are declining. And this is a little bit concerning for scientists like myself and Benoit. And this is one of the reasons we encourage our peers and future generations to make sure to prioritize continued collecting. So what this graph is showing is the number of new additions to collections for each of these taxonomic groups from the 1850s to now. And what you can see is that collections sort of increased from about 1900 to about 1970. In 1970, the rate of new collection has declined for almost every single group. And what that means is that future generations may not be able to answer as many questions as we are able to answer now. So these collections, again, are snapshots of what is happening currently with our world. What is the diversity? What is the size? What is the gut microbiome of these different organisms? And if we don't continue to make these collections, if we don't prioritize continued collecting, future generations are not going to be able to understand kind of these long-term trends that we're seeing. So continued collecting is super important. And again, just kind of coming back to these photos to show that yes, we have a lot, but it's always important to continue to add to that so that we have an improved understanding and that we allow future generations to really understand our world and the changes to biodiversity over time. And that is the end of my talk. And I would love to answer questions that anyone has about ecology or frogs in Borneo or museum collections. Yeah, that's me.
So Jennifer, what's your favorite part as a biologist, ecologist? Going to the field, going to the museum, writing?
Oh, field for sure. I have been, I think like many field biologists, the last couple of years during the pandemic has been really challenging because, you know, my work is in Borneo, but I live in the US. I wasn't able to travel to Malaysia because borders were closed. And so it was, I just felt kind of, you know, I was doing the other parts of my work, like you said, focusing on museum work and writing, but I just have been so eager to get back, and I'm so happy to be back in the field. So yeah, being in the field, and I love animals in general. I love frogs, but I just love all nature. And so being outside, and you know, I get excited when I see birds sweeping on the stream above us, you know, or like cool spiders. You see tons of spiders on the stream, and they're just like crazy big with cool colors, or amazing caterpillars that are like neon green or bright pink, and everything around here. I just love it. I love being outside. I love being in the field. So for me,